This protocol provides a workflow for the production of infectious adenoviral particles that are used to transduce a gene of interest into naive T cells, which, upon ectopic expression of the transgene, are differentiated into regulatory T cells or Tregs. First, the gene of interest is cloned into the adenoviral destination vector. The vector is then transferred into the adenovirus producer cell line HEC293A, and infectious adenoviral particles are generated. The resulting viruses are used to infect naive T cells from mice with an adenovirus receptor transgene. After infection, the cells are then rested to ensure expression of the gene of interest in naive T cells. The T cells are activated in polarizing conditions to induce Treg differentiation. Then, the cells are fixed and stained for flow cytometric analysis. Ultimately, results can be obtained that show expression of the Treg differentiation marker FOXP3 and the infection marker GFP, which may be influenced by the overexpressed gene of interest.
The main advantage of this technique over existing methods, like retroviral gene transduction, is that it allows overexpression of a gene of interest in naive T cells. This results in a high level of overexpression present at the moment of T cell activation.
This method can help answer key questions in the regulatory T cell field. For example, about the post-transcriptional gene regulation that underlies the establishment of Treg cell identity.
Begin this protocol by cloning the gene of interest into an entry vector. The PCR-amplified gene of interest is blunt-end ligated into the topoisomerase-coupled vector p-enter d-topo. The gene of interest is then transferred by LR recombination into the p-cockadoo destination vector. It contains the adenoviral genes and an internal ribosomal entry site, or iris, followed by the open reading frame of GFP to create an adenoviral expression vector with an infection marker. Next, 10 micrograms of the adenoviral expression vector is linearized in a PAC1 restriction digest. The DNA is then precipitated and resuspended in water at a concentration of 3 micrograms per 100 microliters. Linearization liberates the viral inverted repeats, or ITR, which are required for replication and encapsulation of the viral DNA into virus particles. All of the following steps should be performed under the appropriate biosafety conditions for adenovirus infection. To prepare linearized vector for cotransfection, transfer 6 microliters of jet PEI reagent into 94 microliters of 50 micromolar sodium chloride. Vortex briefly. Then, while vortexing, add the mixed solution to 100 microliters of linearized adenovirus vector. Incubate this transfection mix for 15 to 30 minutes at room temperature. Next, remove HEC293A cells grown to 50% confluency on 6-well plates from the incubator. Dispense the solution dropwise into the wells containing HEC293A cells and incubate them at 37 degrees Celsius and 10% carbon dioxide. Add 0.5 milliliters of fresh medium every 3 days. Using GFP expression as an infection marker, evaluate the transfection efficiency visually with an inverted fluorescence microscope. Every 2 to 3 days, check for cytopathic effects, evidenced by areas with enlarged and rounded cells that start to detach. This is indicative of efficient virus generation. Once broader zones of cytopathic effects appear, all cells will be infected within 24 to 72 hours. When all of the cells show signs of cytopathic effects, but before an overall detachment of cells occurs, harvest the cells by gentle pipetting and transfer them with the supernatant to a 15 milliliter polystyrene tube. Next, to rupture the cells, freeze the suspensions on dry ice for 15 to 20 minutes. Then, quickly thaw them at 37 degrees Celsius. Repeat this freeze and thaw cycle 2 more times. The ruptured cell suspension is now referred to as the primary virus lysate. Keep all virus lysates on ice if they'll be used within a day. For long-term storage, freeze them at -80 degrees Celsius. Keep in mind that any additional freeze-thaw cycles will reduce the virus titer by 30 to 50%. To amplify the primary virus lysate, infect HEC293A cells grown to 90% confluence on 15 cm tissue culture dishes with half of the primary virus lysate and incubate for at least 36 hours. After 36 hours, check for infection by fluorescence microscopy. Nearly all cells should be infected. Once all of the cells show cytopathic effects, but not detachment, harvest the cells by gentle pipetting. With efficient virus production, this state is reached within 48 hours of infection. Transfer the cells and supernatant to a 50 milliliter polystyrene tube. Spin cells down at 300 G for 10 minutes at 4 degrees Celsius. Following the spin, remove the supernatant and resuspend the pellet in a suitable volume of medium. 1 milliliter should result in a titer in the range of 10 to the 10th per milliliter. Perform 3 freeze-thaw cycles as before to disrupt the cells. Then, centrifuge at 900 G for 20 minutes at 4 degrees Celsius. After the spin, carefully transfer the supernatant, which is now referred to as the concentrated virus lysate, to a cryotube. Make aliquots for multiple infections and store them at -80 degrees Celsius. Finally, determine the virus titer by FACS analysis of infection marker expression in A549 cells according to the instructions in the accompanying document. This part of the protocol is performed using naive resting CD4 T cells isolated from DO1110 transgenic CAR delta 1 transgenic mice. For small-scale experiments, pipette an appropriate volume of viral lysate to achieve an MOI of 50 into 1 well of a 96 well round bottom plate. Add up to 4 × 10 to the 5th T cells in a final infection volume of 50 microliters in T cell medium. This setup allows parallel experimental approaches. Incubate cells for 90 minutes at 37 degrees Celsius in a 5% carbon dioxide incubator. Following the incubation, spin the cells down at 300 G for 5 minutes at room temperature. After the spin, resuspend them in 200 microliters of PBS. Centrifuge again and take off the supernatant. Resuspend cells in 200 microliters of T-cell medium without stimulating antibodies and without IL-2 or other cytokines. Then, rest the cells for 40 hours at 37 degrees Celsius in a 5% carbon dioxide incubator to allow expression of the gene of interest before activation. To activate and polarize the T cells, pipette anti-CD3 and anti-CD28 coupled beads into a small reagent cup. The number of beads should be equal to the number of cells. Then, tenfold the volume of PBS and place them on a magnet for 2 minutes. Next, take off the supernatant and resuspend the beads in 200 microliters of polarizing medium. Centrifuge the rested cells at 300 G for 5 minutes at room temperature. Following the spin, take off the supernatant and resuspend cells in 200 microliters of polarizing medium containing antibody-coupled beads. Incubate for 72 hours at 37 degrees Celsius in a 5% carbon dioxide incubator without changing medium. The protocol for staining and fixation is described in brief here. First, the cells are washed in PBS and incubated with fixable dead cell staining solution. After another wash step, the cells are fixed in paraformaldehyde, following which the cells can be treated without adenovirus biosafety precautions. After fixation, the cells are washed again and permeabilized with methanol. After another wash step, the cells are incubated with anti-FcR3 to block non-specific binding. Finally, the cells are incubated with anti-FcR3 and PE-coupled anti-FOXP3 antibodies.
There is the risk of losing a lot of cells during the fixation and staining procedure due to the multiple washing steps. To avoid loss, make sure to take off the supernatant right after centrifugation without disturbing the pellet with a pipette tip.
After the cells have been stained, wash the cells twice in PBS and analyze them on a flow cytometer. A 96-well reader on the flow cytometer allows performing T-cell infection, differentiation, fixation, and staining in the same 96-well plate, providing an efficient workflow for parallel analyses of multiple conditions, but the analysis can also be performed in standard fax tubes. To assess the effect of adenovirus infection on T-cell viability and Treg differentiation, naive T-cells purified from D01110 transgenic CAR-delta-1 transgenic mice were infected with control adenovirus at different MOIs and activated in Treg polarizing conditions. Fixed and stained cells were analyzed by flow cytometry for the expression of the infection marker GFP, incorporation of dead cell dye, and for the differentiation marker FOXP3. As shown here, increasing the MOI from 1 to 50 results in a higher percentage of infected cells, while neither viability nor Treg differentiation is affected. To determine whether adenovirus infection affects the activation status of T cells, infected and non-infected cells were analyzed for expression of activation and resting markers by fax. As seen here, expression of these markers was indistinguishable in infected and non-infected cells. When cells were then activated in Treg polarizing conditions, upregulation of activation markers and downregulation of naive T-cell markers was very similar in infected and non-infected cells. Taken together, these data indicate that infection of T cells at a naive state does not interfere with subsequent T cell activation. To test the level of overexpression achieved with the described adenoviral transduction protocol, naive T cells were infected with microRNA 155 encoding or control adenovirus at an MOI of 50. Cells were either rested for 40 hours, activated for 40 hours, or were rested for 40 hours followed by an activation of 40 hours. cDNA was generated and microRNA 155 expression was determined relative to SNO-RNA 202 by quantitative PCR. MicroRNA 155 overexpression levels in naive T cells rested for 40 hours matches endogenous microRNA 155 expression levels after 40 hours of T cell activation. Activation of those cells still induces activation-dependent microRNA 155 upregulation. This demonstrates the high level of overexpression in naive T cells by adenovirus on top of the endogenous activation-induced microRNA expression.
Once mastered, the procedure of cloning, virus generation, and T-cell infection can be completed within 4 weeks. The virus, once generated, can be stored long-term or re-amplified within 3 days.
While attempting this procedure, it is important to remember to carefully control all experiments with an empty vector and to first establish the effect of adenoviral infection itself on the studied process. Please keep in mind that adenoviral infection and overexpression is transient and lasts for only about 5 to 7 days.